As I say, a couple of disclaimers. One is that I'm not a trained coronavirologist. I have 30 years of experience on HIV, and I switched entirely to SARS-CoV-2 when the pandemic started affecting Italy and the US. Obviously, I have ties in both countries, so it became really important. And so I'm a newcomer to the field, but I am a microbiologist and a virologist. And I also have a very nice mask with the coronavirus theme, so I think that adds to it. Now, the other disclaimer that I need to make is that the University of Maryland, with which I have an affiliation, is one of the testing sites for one of the vaccine that I will be discussing, but I have no tie whatsoever with that of other clinical trials. And so, we know the phase of COVID-19. This is data from in Europe. It would be at 3:32 p.m. But we are approaching 30 million people infected. The U.S., India, Brazil, and Russia are the countries most affected by the virus. These numbers are probably all underestimates, because in some places you don't have, you cannot test all cases, but these are good reference numbers as well for the number of deaths. The US is approaching 200,000 deaths, and hopefully there will be some, control of the infection sometimes. Now, as somebody mentioned, there has been a lot of information about coronavirus, COVID-19, etc. In fact, ever since the beginning of the year, from January 1st until last night, we had an average of 200 publications per day, including Saturday and Sundays. So there is a huge output of information. Some of it is likely to be completely irrelevant. Some of it is bad information, and there is some good information. But the information part has really been critical. In fact, everybody has become a little bit of an expert on coronavirus, including folks like this one. And I hope you can hear it.
Because there's no COVID. It's a fake pandemic created to destroy the United States of America.
But the president said to Bob Woodward that there is a virus, the coronavirus, and that it is deadly.
That's his opinion. The truth is that the CDC said that only less than 10,000 people died from COVID. The other 190,000 have 2.6 or 2.8 other mortalities.
So I don't know how much you heard of this, of what this fellow said, that this person was referring to the fact that a lot of people, a lot of the mortality of COVID-19 is observed in people that have pre-existing condition, especially cardiovascular and diabetes. And some people have numerous conditions, and that's for sure a risk factor for deaths of coronavirus. But it doesn't mean that people that have pre-existing condition would have died the day after it had not, if they had not contracted the virus. In fact, this graph that I took from The Economist shows that pretty elegantly, how even when you are getting pretty old and you have pre-existing conditions, and this is the interval to which the person was referring, between 2 and 3 existing conditions. But even when you are above 90, you are really losing 2.5 years of life that you could have lived. And the more you go into the younger population, even with 3 existing conditions, between 80 and 89 years of age, you're losing 5 years of your life. And the toll becomes more and more heavy as you look at younger people. So it is not correct to say that these people have not died of COVID. They have. And there have been a significant number of years lost. So the arguments that we are losing just the weaker, no, it's not true. We're losing them much earlier. We could have them around for much longer. So the SARS-CoV-2, as we very well know by now, is the causative agent of COVID-19. And here is the virus is shown imaged as it attacks cells, lung cells kept in culture. Now, we are very familiar now with the course of the disease. You have an initial phase of the infection where you can have mild or even no symptoms at all. In this phase, our immune system reacts to the viral infection, mostly interferons, which are very good at fighting the virus. In fact, people with good interferon response are probably the people that do not show much symptomatology. So some people have mild symptoms and fever, and they may progress in a second stage, which is where the virus is taking over the respiratory system, the lung. And this is when things start getting hairy, so to speak, because the immune response now, just like a good army, can create damage even to the people that is supposed to defend. Our immune system can contribute to the pathology of SARS-CoV-2, as well as many other microbial diseases, as probably a lot of people on this call know. So, and the disease can evolve further. The cytokine storm and the invasion of the lung tissue by activated cells may progress further. And now we are in what is called the hypoinflammation phase, which is the phase where you may actually, there may be loss of life due to cardiac failure, shock, etc. So, and you have severe respiratory impairments. Now, the one important thing is that the disease is not just while you are in the hospital. A significant number of patients do not have damage only while they are in the hospital, and the damage goes away after they clear the virus. In red here, you see pulmonary damage up to 12 weeks after discharge from the hospital. And in fact, people complain of shortness of breath, weakness, week after being dismissed from the hospital. Not everybody, but still is not necessarily a disease that you clear as you clear the virus. And this is the virus, probably many of us are familiar with this type of figure by now. This is an envelope virus, so it carries with itself part of the viral membrane, the cellular membrane. The spike protein is the one that makes contact with the receptor and causes entry of the virus into the cell. And the genome of the virus is a plus-stranded RNA, complex with nucleocapsid protein, and is 30 kb long. And this is a pretty big RNA virus. So the receptor is an angiotensin converting enzyme 2, which is part of the angiotensin-renin system, which controls several physiological parameters, including blood pressure. And there are proteolytic cleavage that activate the fusion process. Proteases like TMPR, SS2, and furin and big choline into the action of entry. So they are necessary for entry, which is mediated by endosomes, after which the virus genome can be transcribed into protein, and the protein can now the RNA-dependent RNA polymerase can start making copies of the genomic RNA into a minor strand, which then can be copied back into plus-stranded, as well as subgenomic RNAs that are those that allow transcription of proteins, which then are assembled at the endoplasmic reticulum interface and released by exocytosis. And these are some of the drugs that, or approaches that we have to treat the infection, including antibodies, which we will talk a little bit. And there are remdesivir interferes with polymerase. And there are other approaches, including anti-inflammatory approaches and approaches to increase the interferon response. And we can talk about it. The furin cleavage site is a distinctive feature of this virus. And the reason why I highlight it is that furin, unlike other proteases that are important for other viruses, like the SARS coronavirus, of SARS, of 2003 SARS, have a furin cleavage site. And furin is a protease which is pretty ubiquitous. So besides having a receptor, which is the ACE2, which is pretty ubiquitous, in fact, it is expressed in the gastrointestinal tract, in the brain, and there are gastrointestinal and central nervous system. Also, furin is pretty ubiquitous. So this could be one of the factors in the infectivity of the virus. Like with all RNA viruses, we wonder about the mutations that can occur, and the pretty famous mutation at this point, and is the D614G mutation, which seems to impart more infectivity in at least in tissue culture experiments. And this variant seemingly has taken over, become the prevailing variant in several countries. Whether this is a founder effect of the virus that first made it there, or whether this is truly due to a higher infectivity is still under discussion. The data that I've seen in humans talk about potentially maybe a 20% additional infectivity, but is still being debated. There are many reports about that viruses getting weaker or getting more problematic, particularly potentially dangerous mutation ones that seem to make the virus escape antibodies. Thankfully, that mutation is not being seen circulating a lot. And the good news about even the 614G mutation is that it's still, this virus is still susceptible to neutralizing antibodies. A quick word on how the virus causes pathology. As I said, most of it is mediated in the immune system. As the virus enters the alveoli in the lung, it starts becoming cytopathic, and the cell death signal can activate macrophages. There are many other signals that can activate the macrophage, and some of them come from NK and activated T cells, which further activate the macrophage. The macrophage itself has the ability to identify viral infection using specific receptors. And there is another potential component to this, which is against the antibodies, okay? So antibodies that are non-neutralizing may bind to specific receptor on the macrophage, so-called Fc receptors. So this is a property of antibodies. Typically we see antibodies as blocking the pathogen, and this is what we call neutralization. But antibodies have a number of wonderful reagents that have a number of effector functions, so-called, by binding to so-called Fc receptors. So the macrophage is one of the cells that expresses Fc receptor, and binding Fc receptor of antibodies may activate further the macrophage. And there is some question whether macrophages in the lung may be activated to actually express ACE2 themselves, and that's being debated. But, so there are several mechanisms that activate the macrophage, and these causes the macrophage to become activated, start producing cytokines, and that is part of the induction on the cytokine storm. So IL-6, TNF, and other cytokines are really prominent in COVID. Another part of the equation is together with the macrophages, there are other cells that are involved. One is the neutrophil. And there is also a role of the endothelial cells. Endothelial cells express ACE2, so they can be infected. And some people are thinking that this may actually be a very strong participant to the pathogenesis. So the endothelial cells can activate the neutrophil to release so-called neutrophil extra traps, extracellular traps. So these are when the neutrophil starts spewing out its own DNA. Part of the activity is to stop bugs like bacteria, by making everything denser, but some of it can trigger coagulation pathways. Other coagulation pathways are triggered by the macrophage. So this is probably part of the reason why you have massive coagulation in some of the patients. And these are the nets, these neutrophil extracellular traps, which are revealed in this micrograph. When they are revealed, specifically when serum of COVID-19 patients is added to the tissue culture. But going back to the vaccine and neutralizing antibodies. But that's the type of antibodies that we want to induce. We don't want to induce potentially other antibodies that can activate the macrophage. So it's very important in the context of vaccines to make sure that we have the right antibodies. That's part of the safety. So of course, the question is, can antibody protect? Thankfully, there is strong evidence, clear evidence that they do, and this is I'm not going over all of the evidence, but this is one study that is very informative. It is one of the earlier studies with an fishing vessel where they studied 122 people before and after a significant COVID outbreak. So out of the 122 people that were studied, 104, 82.5% became contracted COVID during the study. So some of since there were data from before the outbreak, the antibodies were detected in 6 of the people that were studied, and in 3 of them, the antibodies were actually of the neutralizing type. And this is the activity or the neutralization activity of the antibodies. Non-neutralizing antibodies don't have that kind of activity. So, and this seropositive with the neutralizing antibodies prior to the outbreak remained negative. They did not have COVID symptoms during or presence of virus during the outbreak, showing that they were strongly protected from the infection. Also in animal models, there are several animal models for COVID. One of them is the macaque, which has some level of symptomatology. So when macaques get infected, they do have virus. But if you take the same macaques and try to re-challenge them with virus, now you do not see presence of virus. So this is very clear evidence that they get protected. Whether it is only antibody or whether this is a very short term between the first and the second challenge. So there may be something else working here, which we can talk about it after I'm done with this segment of the presentation. So these macaques have very good titers of antibodies, and there is some titers of neutralizing antibodies. So this is good news. This is good news for the development of vaccine. There are a lot of different approaches, and we can talk about some of them, but in the data that I'm going to talk today, I'm going to talk about RNA vaccines, which are very easy to produce, and in fact, they are currently at the forefront of vaccine production. And as you probably all know, there are several phases in the clinical trials, from safety, phase 1, extended safety and efficacy, phase 2, and efficacy, phase 3. And this is the current number of phase 1, phase 2, and phase 3 clinical trials that are ongoing in the world. Some vaccines have been given limited approval, mostly in Russia and in China, meaning that these vaccines are given to a segment of the population while they are doing phase 3 trials. I'm not sure that that is the most expedient way to do things, but that's what's going on. There will be terrible logistics problems in deploying these vaccines, but that's not something that we can do much about it as scientists. So there is currently an ongoing phase 3 trial of as I say, of mRNA vaccine, this so-called mRNA 1273. There have been already a phase 1 trial, which is still ongoing, so on 120 patients to test different doses of the viruses. And then there is an ongoing phase 2 clinical trial, which has shown that overall this vaccine is safe. And this was divided into 4 arms, different doses, and also different age groups. And so far the news are all positive, and that's why this is progressing to a phase 3 trial with 30,000 subjects being enrolled, 1 dose, experimental or placebo. And there will be 2 injections at day 1 and day 29. So looking at the data that have been published, this is again in the macaque model, which is limited disease, but these macaques, as you see at the higher dose, produce very nice titers of neutralizing antibodies. At the lower dose less, and as you see, individual monkeys, as is expected, express different levels of neutralizing antibodies. After the second vaccination, the titers become very good in the monkeys. They don't give absolute protection. You still seem to detect a virus in the monkeys after challenge, at least in some animals. So it's not 100% sterilizing protection, but the monkeys have no symptoms. So they are effectively protected from disease. This is a part of the data instead from a phase 1 clinical trial in humans, where there are very good concentration titers, as they are called, of antibodies that binds to the receptor binding domain of the spike protein. And when you test them in a viral infectivity assay, they do neutralize very well the infection. So the data for now are very good, and that's why the vaccine is progressing into a phase 3 trial, and which will take some time to perform, and to get some information on the level of antibodies and potential outbreak infections. One word of caution is that we'll have to keep monitoring for 2 things. One, as I said, is the antibodies that may be not so good, the antibodies that may activate the immune response, and we don't want these antibodies to be induced by the vaccine. But also, this is an experiment done by the Barik group at UNC, and this is with SARS vaccine. This is not, so this is many years ago, about 10 years ago, where they tested the efficacy of vaccines in young versus old mice. And the vaccines seem to be losing efficacy in the old mice. So another word of caution is we'll have to make sure that we are protecting the people that need to be protected, which is the most vulnerable are people with pre-existing conditions and elderly people. So have we learned something from COVID? I would like to say so, but I'm not sure. And the previous speakers also touched on some of these aspects. One is, are we going to have more pandemic readiness? That would be really nice. And I was really thrilled by one of the previous speakers that was telling us how in Brazil, they are seeing a lot of viruses in the animal population that may be ready to come onto humans. So before to this pandemic, we were really concerned about influenza, and we should still be concerned about influenza. But we should also keep an eye on all the emerging viruses that are out there. Hygiene obviously plays a big role in prevention, and I hope that we are learning to impose stringent rules for agriculture, keeping livestock and food markets, you know, because the more humans and animals come together, the more you have the chance of viral transmission. More attention to environmental issues, because the more we change the climate, the more species that normally are not in an area move to that area. And so now you have increased chances of transmission. You may have more favorable conditions for transmission, etc. And finally, safety versus speed. We'll have to have a debate, and we're not going to have it today, but how fast do we want to go with releasing, decreasing the level of attention to and preventative measures like masks, etc., lockdowns, in order to recover, have the economy recovering, in order, and how fast do we want vaccines to proceed to deliver them to the population. So these are things that we need to think about. And I took longer than I wanted to, but this is what I wanted to say to introduce the topic, and I'm happy to discuss more any of the aspects that I touched upon. And
thank you very much. That was fascinating, and has taught me a lot in these last 30 minutes. But you've also been helping us to keep our community up to date, you know, for the last 9 months, to be honest, because I remember we were drafting statements back in January, to put on the FEMS website. But before we go into that, I just wanted to ask a couple of questions. So in this case, we're looking, you had some nice data about how rhesus macaques were able to, what's the word, kind of resist infection.
Yes.
After being, I guess, inoculated.
This was one of the best news of the year. You know?
Yeah. But has, since this been published, anyone repeated it to see if, you know, the immunity can hold over time, or, you know, so I mean, one of the things I remember reading was that coronaviruses are quite hard to make a vaccine for that gives you a long span of immunity.
Okay, very good point. Duration of effect. How long do these antibodies stay around? So a couple of considerations. One, yes, as I showed, you know, some evidence of protection in monkeys has been shown also with the vaccine candidate. I showed this data in another slide. If I did not highlight it enough, you know, this is the vaccine that is being tested in one of the phase 3 clinical trial. These are data not from human, but from the macaques, you know. And so you see protection, maybe not sterilizing protection from infection, but you see protection from symptoms, which is nice. And so, I think this data is being replicated. The other thing is that one thing is the natural infection, and the other thing is the vaccine, okay? So how long is the antibody protection in a natural infection? People these days are willing to say months, okay, but they're not. The fact is, we have not even had 1 year of a complete cycle of this pandemic. So we do not. The only way to know the final answer, how long the antibodies hang around in a natural infection, is actually to study natural infection for several years and in a big enough sample of patients. Yes.
Because that's one of the things. It's, you know, it's impossible to answer the question, do you retain immunity 4 years down the line? Because we haven't had 4 years since people knew of the virus to be able to do any test like that. No. And I still find it bizarre to think it's only September of this year I mean, it feels like an endless amount of time, but yeah, it's been probably less than a year since anyone really would have been discussing this virus, much less than a year that the public would have known about it. So there just hasn't been the time to learn everything we need to know.
Exactly. And there are proper ways to do these things, but the only proper way to assess the half-life on antibody response is to assemble a cohort of patients and follow them in time. Bear in mind the fact that some of them may get re-exposed to the virus, you know, so there may be confounding factors. And then again, we already know that some people got infected the second time. So what happened there? Well, in one of the cases, we don't know because we don't have enough data on the patient that got reinfected. On the second patient that I know about that got reinfected, it appears that this patient did not make a lot of antibodies. So, and we know that, you know, we know that people, probably the reason why people get severe disease is because from the onset, they don't make enough interferon. And then when they get into the disease, you know, there is a lot of lymphocyte death, you know, so probably some people are not able to mount an effective antibody response.
Because one of the questions I also had was, back in March, I was ill with coronavirus-like symptoms, and I was off work for about 10 days, and it took me 2 weeks to recover. And at that point in the Netherlands, they weren't testing anyone who wasn't in a vulnerable category because it was right near the beginning. They didn't have the testing capacity. Is there any way that I can get a test of my antibodies? Or there's no real procedure for this, but would an antibody test of a certain kind be able to inform me if it had been real?
Right now there is a plethora of antibody tests, you know. So I do not know what is the policy in the Netherlands specifically. If you can ask your doctor to be tested, I would have to look. I mean, you can probably find that information much faster. But yes, the tests are widely available. Of course, there is a prioritization of if you suspect that you've been infected, you might still have antibodies if you mounted a good response, and they may be measurable, you know. And you know, I suspect they should, you know. And in fact, it's like in the case of the fishing vessel that I spoke about, you know. These people were studied retrospectively, and they find out, no, these people had antibodies, you know, so you can find them for sure. Okay.
One other thing I want to do is just, if I can share my screen, which is to show you, I think I can do it here, a little bit of data on how you changed FEMS's website statistics. So, I mean, you helped us put together this expert statement for kind of more of a lay audience back in March. But one of the things that it did was this spike here, and if you look at this is all normal web traffic, is entirely down to this expert statement. So it kind of completely changed the entire year's worth of website traffic to our to the FEMS website by drawing in so many people across all the different languages. And this spike here is International Microorganism Day, and still hasn't quite reached the peak of the coronavirus information frenzy that was the middle of March and the beginning of April. So there's now, I find this quite bizarre in the sense it's like a record of human behavior caught in website statistics. And then if you look at here, all the physical events in the world were cancelled, and so actually traffic to the website went down. But this is a spike due to your your writing, so I think that's down to you.
I mean, it is impressive, and in fact, that spike corresponded also to a number of invitations to interviews, you know, by different news outlets, you know. Obviously people wanted to know.
Yeah, and the different languages you can see kind of ranked here in the different translations we had. But this is the interesting slide in terms of where people were coming from to access this information. And we were putting it out in different languages. So the ones that were written in Serbian and Italian and Portuguese and German were actually the most well viewed, because I think, you know, lots of stuff's going out in English, but if your English isn't your first language, and, you know, complicated scientific information also isn't your forte, then having something in your local language is super helpful. So yeah, lots of good information being delivered here at a crucial time.
Yeah, well, happy to contribute a new statement if you think is.
Yeah, I think the antibodies would be an interesting extra element. Do you know what I mean? So that was so unknown at the time, and a bit more work has happened now.
Okay.
I think we've lost your webcam.
Well, actually, I think you turned me off.
Oh, did I turn you off? Sorry about that. There we go. I think I tried stop sharing and I ended up stopping all your video. I'll put my apologies.
Okay. I don't think I'm that handsome, that people should look at me. Maybe it's better they don't. But anyway, so yes, I mean, the fact is, I shot that news clip, on purpose, because I think it really delivers the message that, by the way, other speakers were talking about before, the fact that you have all this bad information out there, these beliefs, and these one-sided, like, I think really the point about the mortality is really a key one, because there are these conspiracy theories websites that tell you, oh, people die of other causes. No, they were not supposed to die.
Yeah.
You lost your grandparents way before you should have. And, I lost my dad when he was 91, a few years back, and God knows, I could have used another year with my dad, even if it had been one,
Yeah. And I think if you're the one who's suffering, there's no point in which you're like, oh, well, I only had 3 years to go. That doesn't.
Sorry. And the point I raised about the symptoms that don't go away when the virus goes away, these are things that one of my relatives, she's 30 years old, she's a cardiologist, and she contracted the COVID in the hospital, and she was tired for weeks after the onset of disease and after clearing the virus. So these things need to be out there, and we should publicize them, because it is important that people have good information.
Yeah. Well, I hope this can be part of that effort. And this is all being recorded. So we're going to cut these long live streams into the individual videos. So you have the name of the speaker and the topic of the talk, and they'll all go up on the IMD YouTube channel.
Well, thank you for organizing these. I think we should have more days like these. We should make people more aware of microorganisms. I also take FAMS and the Portuguese Society for Microbiology. Great job. Thank you.
Awesome. Well, thanks for joining us. Our next speakers are on the way, but it's been great, and you've taught me many new things. And over the next year, all the videos will be going out across to try and fill the gap between today and the next International Microorganism Day. So hopefully we can keep content going throughout the year. So great to chat to you, and enjoy the rest of your day. It's the morning.
Good evening.
Yeah, it's the 6:41 in the evening here. But enjoy your day.